Hey everyone, today we're going to talk about microscopes. We're going to talk about the different types of microscopes, just kind of an introduction to them, how we care for them, the parts of the microscopes, as well as the types of microscopes that we have in class available to us. So the first microscope we're going to talk about is called the light microscope. This is the one that's most commonly found in schools. What it does is it uses a light source and it magnifies the image, whatever is on the slide, through lenses. So it uses lenses to magnify an image. Some of the common magnifications would be 40 times, 100 times, and 400 times. So what that means is what the object appears to be 40 times bigger than what it actually is, or it appears to be 100 times bigger. Oil immersion lenses can be available. That gets the magnification power up to 1,000 times. So that's kind of the maximum of the light microscopes. Another type of microscope is called the stereoscope. This allows you to see 3D images. It does not magnify near as high of power as the light microscopes, but you can view things that light can't pass through. So like an insect. You can even stick your finger there and see like what your finger looks like under some magnification power. And so it creates a 3D view. The microscopes really create a 2D view. Okay, and then we have electron microscopes, and the first type of electron microscope is a scanning electron microscope. This allows you to see 3D specimens. You can see a 3D view. You can magnify up to 2 million times. So these are much higher magnification powers, and so we can see a lot of different things here. We can see an ant here, different pollen, things like that, mouth parts of an insect, and so we can see these different images, and they can be magnified. The thing is that whatever is magnified with a scanning electron microscope, it does have to be dead. It can't be alive. The light microscope can actually view living things a little bit. The other type of electron microscope is the transmission electron microscope. This uses 2D, it produces 2D images, but the images that you see it come from very thin slices of something. So actually they take a cell and they actually slice a cell, kind of like you would slice a loaf of bread or something like that, and they take a very thin slice of that cell, and then they get an image from a transmission electron microscope. And what happens is electrons actually go through the specimen. And so what you see with this is an image like this. So you can see we have a cell here. We can see different parts of the cell. So we have a nucleus, and we can see all these other organelles that are in a cell as well. So this is what we can see a little bit more with that. The different types of microscopes can have different types of light sources, and so you can get some different images. We can also use different types of stains to get different images. We can see different things on the cells, and so these are all cheek cells. These are all human cheek cells right here, and so different types of light and stains, we can actually see different structures within those cells. So we can see kind of the outside here, that's the cell membrane, this little dark circle in the middle, that's the nucleus, and so you can kind of see there's the nucleus in this one. This one's a little bit folded over, but you can still see the cell membrane on that. So we can see the different cell parts a little bit differently and easier under different stains and different light sources. So when we use a light microscope, you always want to carry a light microscope with two hands. We only use lens paper for cleaning. We don't want to scratch the lenses. You don't want to force anything when you're using a microscope. You don't want to force the knobs. Well, we store it when covered. We store it covered, and we keep objects away because you don't want to trip over cords and things like that. Okay, so big thing on the light microscopes, when we carry them, two hands, we don't want to force anything. Everything should move easily. If we force something, we're likely to break something a little bit more. Okay, so we have some microscope parts here. We have the eyepiece, which is at the top. That's also called the ocular lens sometimes. We have the arm. That's what you would carry it with. You put one hand on the arm and one hand on the base. We have the stage. That's this black kind of square looking thing. That's where we stick our microscope slide. The coarse adjustment knob, that moves the stage a lot faster, and so you use it to focus at low power. And you have the fine focus, and that moves it in really small increments, and so it's a little adjusting your focus. You have the base, and so that's the other part we carry. So we put one hand on the base and then one hand on the arm, and that's how we carry a microscope. We have the body tube. That's just the area that we look through from the eyepiece. We have what's called the revolving nose piece with the different objective lenses. And so these different lenses, we can just turn this. We can turn that. It just turns really easy. And you can move from one lens to another, from one magnification power to another. You have stage clips that hold your microscope slide in place. We have a diaphragm that That it determines how much light passes through, and then we have our light source down there at the bottom. So the magnification powers of a microscope, what it is the eyepiece or the ocular lens. So if you remember, we have the eyepiece right here. This is always what we call a 10 times magnification power. And then these three lenses right here, the objective lenses, most of the time they're either 4 times magnification, 10 times, or 40 times. And so to get the total magnification power, we take whatever our objective lenses are. Say we're on the 4 times. The ocular is always 10. We take 4 times 10, and that's the total magnification power is 40. Or if we're on the low power, which is a 10 times, we take 10 times the eyepiece, 10, and we get 100 times magnification power. Or if we're at our high power for our microscopes, which is 40 times, we take 40 times 10, and you can get to the 400 times magnification power. And for our microscopes, that's really kind of the maximum that they have. So using the microscope, general procedures, you just want to make sure everything's cleared out of the way. The cords are out of the way. When we carry it, we have one hand on the arm and one hand on the base. To focus specimens, we always start with the scanning objective. The odds are you'll be able to see something on this setting, and so you want to use the coarse adjustment knob to adjust it to get it into focus. But then you can switch, after you get that into focus, then you can switch to the low power. And once you switch to the low power, you can maybe use the fine focus. If you have to, you may have to use a little bit of the coarse focus knob, but then you can use the fine focus to get the focus just right, and then you can switch to the high power. But at the high power, you only want to use the fine focus knob. You don't want to use the coarse because it moves it too quickly, and the lens is actually going to be really close to the slide, and if you move it the wrong way, you could actually run the slide into the lens and then break something, and so we don't want to do that. And so you don't skip steps. So you always start at the scanning, and then you move to the low power, and then you move to the high power. Okay, so you must focus on the low power before moving to the high power. Okay, you don't start at the longest lens of the objective of the objective lenses. Okay, you don't want to start with the longest. You want to start with the shortest. Okay, so you start with the shortest, which is low power, and then you move up step by step. Okay, finally, when we're drawing what we see on the microscope, we want to label things as best we can. So like here we have an amoeba, where we would actually label the cell membrane and the nucleus because that's what we can identify, and then we label at the top what we are looking at and what power we're looking at it under. And so we can see the different size, the relative sizes of them, and so we can see that. And so our labels, we want to write them as clear as possible. We want to label things as best we can when we do these microscopes and look at these slides. All right, guys. So now you're going to be working on a little assignment that identifies the microscope parts as well as cell organelles. All right, so like always, if you need any help, don't feel free to ask. Don't hesitate to ask, okay? All right, have a good week, guys.